The problem we are focusing on is soil contamination caused by polyaromatic hydrocarbons in the municipality of Yondo. Yondo is a municipality in the subregion of Magdalena Medio, in the department of Antioquia. Why did we choose Yondo? We chose it because the hydrocarbon industry is one of its main sources of employment, and because of the mismanagement of the oil companies or due to criminal acts, oil spills occur.
Bioremediate soil contaminated with polyaromatic hydrocarbons using in vitro cultures containing Pseudomonas aeruginosa and Bacillus subtilis. First, extract and identify soil contaminated by PAHS by means of liquid chromatography. Isolate and characterize by means of microbiological culture the bacteria Pseudomonas aeruginosa and Bacillus subtilis from a sample of soil contaminated by PAHS. Establish mixed liquid cultures with the bacteria Pseudomonas aeruginosa and Bacillus subtilis to bioremediate the soil contaminated by PAHS. Evaluate the efficacy of the process, the bacteria Pseudomonas aeruginosa and Bacillus subtilis, by means of HPLC and cell count.
Environmental impact is a local level, since soil is a finite resource, which means its loss and degradation is not recoverable in the course of the human lifetime. Soils affect the food we eat, the water we drink, and the we breathe, our health, and all or the organisms on the planet. Without healthy soils, we would not to be able to produce our food. It's estimated that 95% of food is produced directly or indirectly in soils. The bioremediation we are going to use is different because it doesn't create waste in quantity and toxicity, unlike other methods, and it does not affect the soils, and he has few requirements. Because it's a very fundamental biotechnology strategy to remove the contamination in soils throughout other life beginnings, such as or bacteria, which allows a general protection to human and health and other living beings. Is environmental friendly, is not expensive, and is very feasible.
The methodology that will be used for the elaboration of the project is adapted to the condition of the equipment and laboratories provided for this purpose. With this, it deserves to express in details the steps to follow experimentation. In Yondo, Antioquia, the process of collecting soil samples will be carried out and composite samples will be made. Then a quantification analysis of polyaromatic hydrocarbons will be carried out by means of a liquid chromatography. After the soil sample, we will isolate and characterize the bacteria using culture media as a nutritious broth and then a nutritious agar. Then a characterization will be made by a selective agar determining Pseudomonas aeruginosa by a streamide agar and Bacillus subtilis by means of a nutritious agar.
Subsequently, a macroscopic and microscopic characterization of the bacteria is performed using optical microscopes, where the following structures are identified: Bacillus subtilis and Pseudomonas aeruginosa. From these collected soil samples, serial dilution will be made in order to perform a progressive reduction, step by step, of the concentration of bacteria and colonies present in the soil sample. These serial dilutions are used to reduce the concentration of other types of bacteria or external agents in the sample. After this process, a gram stain is performed to determine whether the bacteria is gram-negativa red stain or gram-positiva blue stain. For efficiency, take sample of the culture of Pseudomonas aeruginosa and Bacillus subtilis or nutrient agar to see the growth of both bacteria.